Science Guy. Bill Nye, the Science Guy. Bill Nye, the Science Guy.
Science rules.
Bill Nye, the Science Guy.
Hi, I thought you guys were coming today. Come on in, we're doing some great experiments in the lab. Nice to meet you. I'm Bill Nye.
You're not the real science guy.
No, I'm not the real science guy, but I am Bill Nye's clone. Now, a clone is an exact genetic replica of Bill Nye, so I'm Bill Nye.
Cloning has always gone on in nature. Identical twins are individuals that split from the same egg and are thus genetically identical. Likewise, with self-pollinated plants, the daughter plants are genetically identical to the parent plant because only one set of DNA from the parent went into the new organism. Even worms, when cut in half, regrow into two separate but identical entities. In the late 1800s, Hans Dries sparked interest in experimental cloning through his research with sea urchins. Dries took a two-celled embryo of a sea urchin and shook it in a beaker full of seawater until the two cells separated. Each grew independently and formed a separate whole sea urchin that did not lose any genetic information. After experimenting with salamanders, Hans Spemann successfully transferred the nucleus from an embryo to an empty cell. The cell eventually developed into a normal salamander. In 1902, he presented the idea of what he called a fantastical experiment, in which the nuclear material from an adult cell could be taken and transferred to another in order to create a genetically identical copy. In 1951, Robert Briggs, along with some contributing scientists and frogs as his test subjects, performed the first successful nuclear cell transplant with the use of embryonic cells.
Now that you know a little bit about the history of cloning, my lab assistant here, Val the Science Gal, is going to tell you a little bit more about how cloning is actually conducted.
Thanks, Bill. Now you see, you have a donor egg, a cell for the intended use of a clone. You take the genetic material out of the donor egg. All that's left then from the cell is the cell remnants besides the DNA. Then you have a somatic cell.
A somatic cell is any cell that isn't either an egg cell or a sperm cell.
Now this DNA is intended for the clone. You take the DNA out of the somatic cell. Now I have the DNA in my pipette. I can then take this DNA intended for the clone and put it in the cell of the donor egg, as seen here. We now have a cell with the donor's DNA for a clone. Once the cell is made, it is treated with an electric shock in order to stimulate the growth of the cell. The cell is then able to grow into multiple copies and make a blastula. These are all identical copies of the intended cell for the clone.
This process was used to create the first cloned mammal, Dolly the sheep. While Dolly was a success and was able to reproduce herself, her lifespan was half that of a normal sheep, and she possessed further health issues. Because of severe arthritis and a progressive lung disease, she had to be euthanized at the age of 6.5.
This hotly debated topic has its fair share of arguments coming from both viewpoints. Those who are pro-cloning say that growing organs from a person's own tissue via this process reduces the likelihood of that person's body rejecting it, since it is genetically identical to the patient's own one. Cloning can be used to manufacture stem cells, which in turn can be used to grow organs for transplant as well. In addition, cloning is a very viable solution to treat infertility and allow the mother to still produce a child biologically related to her. Other beneficial uses include making plants immune to diseases and creating cows who would be able to produce different medicinal types of milk. However, there are many downfalls to this process as well. For instance, all of the moral and religious issues that arise. It also has the potential to lessen our world's beautiful diversity and may weaken our ability to adapt. So far, the research done in cloning has had very few successes. Only 2 or 3 in every 100 clones are actually born alive, and those few that are turn out to possess several genetic problems. These genetic abnormalities are solely brought about by genetic manipulation by humans and are never seen in the natural world.
The blood vessels, the skin, the musculature of the body
defects,
wall.
heart defects, limb abnormalities, and facial abnormalities.
Have been born with their insides on the outside. We know that they have immunological problems, they have pulmonary problems, they have cardiac
And then
problems.
within hours of birth, they tend to die.
The musculature of the body wall, the brain, the face.
Some of them
I think
are able to breathe, but only breathe if they're on oxygen. Who ever heard of a calf grazing with oxygen on its back?
There are so many misconceptions that occur with cloning. For example, this video shows a very common misconception.
Sorry, you go.
What exactly is wrong with this video? In reality, there are many years between the cloned organisms and the parent. The cloned organism just has the same DNA as the parent, nothing more. Another misconception cloned organisms have are that they are merely test tube creations. In reality, the cloned organism is placed and developed in the female's womb, similar to what happens naturally. After Dolly was born in 1997, people assumed that scientists would soon be cloning humans in no time. Frankenstein was about to become a reality, and this hype caused a lot of fear and refusal to support any type of cloning. Cloning will cause the clones to form up their own army where they all think alike, are all conformists, and that children may be bred for spare parts. In reality, clones are just genetically identical individuals, just like identical twins. Identical twins do not act exactly the same, so clones do not act the same as their parent.
Now that you know the history of cloning, the general cloning process, and some generally accepted pros and cons of cloning, let's go around the globe and get some personal opinions from people on the street.
I feel like cloning is ever growing in popularity here in the United States, and I feel like it's something that is present in some of the less organized organisms right now, but it will be more present in more animals and maybe even humans in the next couple years here soon.
I'm for human cloning in terms of taking cells to kind of, produce like a liver or a heart. If like for organ transplants, for example, it's a really good thing, I think, because there's not enough organ donors. So I think growing a tissue in culture is better than having like somebody go without a liver and passing away because of that. But I'm not for human cloning in the terms of actually growing another person from somebody else.
I really don't agree with it because I feel like everybody should be unique and created the way they're supposed to be.
Well, personally, I would have to say I'm against the idea of cloning, more for the religious aspect of it. I don't believe that, humans should be allowed to neither create nor destroy life.
I don't think they play a huge part.
I would say they do change. I don't know whether that makes me a hypocrite or not, or like whether I go against my own ideas, but I believe that plants, it's all right, but like when you get into like animals and humans, I don't really think it's necessarily a good thing.
Well, like if it was a puppy, I'd really want to clone it if I like the puppy, and like if it was humans, like if I like the person, like if it was me, like I want more of me. If the flower was like a pretty flower, then I want more of it.
Now, with all of that said, the important thing to consider about cloning, like with any other science, is that cloning is a tool. It's up to us as a community to determine how we want to use cloning and to what extent we want to use it. It's up to us to consider whether or not cloning is ethical or not.